In order to understand how dialysis works as a nurse, and to solve questions 20 and 21 in Chapter 9, we need to understand how diffusion works and what it is. So, imagine we have a container, like we do here, and there are two parts of the container that are separated by a valve. That's that gray line there. So, we have 4 different atoms in the container on the left, different colors, and if we suddenly open the valve, let's just think in terms of probability, how many ways can all of the atoms stay in the one container? There's only one way. They all have to stay in the container. Okay, now how many ways can you have 3 atoms in the container on the left and 1 in the container on the right? There are 4 ways. You could either have the orange move to the right, you could have the purple move to the right, you could have the yellow move to the right, or you could have the blue one move to the right. So there are 4 ways that you could have 1 molecule on the right container and 3 on the left. Notice that that's more ways than for them to all stay on the left. In other words, there are more ways that these molecules can spread out than there are ways that they can stay in one place. So, molecules tend to spread out. If you think about that same scenario, but all of the atoms being the same type, you would end up with atoms as spread out as possible, because there are more ways that they can exist. There are more ways that they can end up spread out than there are ways that they can stay all in one place. The tendency of atoms to spread out, because there are more ways that they can spread out than that they can stay in one place, the tendency of atoms to spread out is called diffusion. And it's responsible for so many things that happen in biology. It's the reason why if you put dye in water, like a drop of blue dye, the dye spreads out in all of the water. And it never happens that dye that's spread out suddenly comes back into a single drop. It's the reason why if somebody sprays cologne in one end of the room, and it's only in that end of the room at first, eventually it spreads out to the whole room. This sense of diffusion, molecules spreading out because there are more ways that they can spread out than that they can stay in one place. We can use the principle of diffusion to clean people's blood, and that's dialysis. So let's say instead of a container, we have a tube. And the tube has tiny, tiny holes in it. So tiny, tiny holes. Let's do these. So there are holes in this tube. Now these are microscopic holes. So you wouldn't be able to see them with the naked eye. With the naked eye, the tube would look perfectly solid, but microscopically there would be these small holes. And then let's say there are some things in people's blood. Some things, like the orange dots, which are small, and some things, like the pink dots, or let's say the blue dots, which are large. Now what we can do is we can put this solution, and this would be, inside this tube is meant to represent somebody's blood. With the stuff dissolved in it, like the orange dots and the blue dots. And then outside would represent just some water that we dunk this tube in, just pure water. Another way of saying pure water is distilled water. Now, these molecules in the tube are going to want to spread out. So the orange dots would naturally move out of the holes in the container and spread out, and that's one way we can clean those molecules out of a person's blood. The blue dots would also naturally tend to spread out, but the holes in the container are too small for them to fit through, and so they stay in the container. Anything that stays in the container is called a colloid. Colloids are things that are too big to move from a person's blood to pure water through diffusion because it can't fit through the holes. So this container is called a dialysis bag. These walls here, would be a dialysis bag. And the process of cleaning somebody's blood by letting small impurities escape through tiny microscopic holes while keeping important molecules that the body needs, that is called dialysis. So the idea with dialysis is that you will eliminate things that are very small from somebody's blood, and you won't eliminate colloids. Okay, given that context, let's think about these two questions. So question 20 says, each of the following mixtures is placed in a dialyzing bag. That's that tube that we just drew with the tiny little holes in it. So that's the dialyzing bag. And immersed in distilled water. And we saw how before you could take this and have water on the outside. Which substances will be found outside the bag? And the answer is anything that's small enough to fit through the holes. Anything that is not a colloid. Now in these questions, they tell you what things are colloids and what aren't. So if something is not listed as a colloid, it will be outside the bag. In question A, they do not use the word colloid. And so these things would be like the little orange dots, and this would be found outside of the bag. So you can write NaCl is not a colloid, and so would fit through the microscopic holes in the dialyzing bag and would be found outside the bag in the distilled water. Beautifully clear explanation. Okay, now for B, we have two things. We have starch, which they tell us is a colloid, so that's not going to be outside of the bag. And then they give us alanine, and alanine they don't say is a colloid, so that will fit through the microscopic holes and be found outside the bag. So you could say starch is a colloid and won't be found outside of the dialyzing bag. And then for alanine, you can take the same explanation that we have above, but we'll just change it from being NaCl to being alanine. So you can take that and actually, well, yeah, okay. So we'll fit that here. So you would have the explanation that instead of NaCl, you would say alanine is not a colloid, and so would fit through the microscopic holes in the dialyzing bag and would be found outside of the bag. So alanine would go outside of the bag, but the starch would be too big and it would stay stuck inside. All right, NaCl solution and starch solution. So with this one, they mention NaCl, and they don't say that that's a colloid, so that's going to go outside of the bag. And then they give you starch, which is a colloid, and so that will be staying inside the bag. So you could say that starch, so you could use a similar explanation as before. I'll just copy and paste this. And what we can do is just change the molecule. So you'd be writing a similar explanation. Starch is used here again, so we could say starch is a colloid and won't be found outside of the dialyzing bag because it's too big to fit through those holes. NaCl is not a colloid, and so would fit through the microscopic holes in the dialyzing bag and would be found outside of the bag in the distilled water. All right, how about a urea solution? They don't say this is a colloid, and so it will be small enough to fit through the holes, and it would be found outside of the dialyzing bag. So we can take our explanation. We only have the thing that is not a colloid, and we're not dealing with alanine, we're dealing with urea. So what you could say for this, what you could write for this is that urea is not a colloid. And so it would fit through the microscopic holes in the dialyzing bag and would be found outside the bag in the distilled water. Okay, 21, similar thing. It says each of the following mixtures is placed in a dialyzing bag like we saw before, and so that would be just like this here. Placed in a dialyzing bag and immersed in distilled water. Which substances will be found outside the bag? Anything that's not a colloid and so small enough to fit through the microscopic holes in the dialysis bag. So they give us KCl and glucose solution, and notice that they do not list those as colloids. And so you could say both KCl and glucose will be found outside the dialyzing bag because they are small enough to fit through the microscopic holes in the dialyzing bag, since they're not colloids. Because colloids are molecules that are too big to fit through those holes. Okay, for B, they say albumin, and they tell you it's a colloid. Because it's a colloid, it will not be found outside of the dialyzing bag. It's too big to fit through the dialyzing bag. So you'd write albumin will not be found outside the dialyzing bag because it is too big to fit through the microscopic holes in the dialyzing bag. And we know that because it's a colloid. So, since it's a colloid. Okay, for C. C says an albumin solution, which, because they know it's a colloid, will be too big, a KCl solution, and a glucose solution. So if you had all three of those together, well, we saw each of those individually in the previous problems. So the first thing we know is that the albumin will not be found outside of the dialyzing bag. And we know that because it is too big to fit through the microscopic holes in the dialyzing bag since it's a colloid. The KCl and the glucose, on the other hand, those would be found on the outside of the dialyzing bag because they are small enough to fit through the microscopic holes in the dialyzing bag, and we know that because they are not colloids. All right, last but not least, we have D. D says a urea solution and an NaCl solution, but notice how they don't list either of those as a colloid, and so both of them are small enough to fit outside of the dialyzing bag. So the answer will be similar to A, except instead of KCl and glucose, it will be urea and NaCl. So both urea and NaCl will be found outside the dialyzing bag because they are small enough to fit through those microscopic holes in the dialyzing bag. And we know that because they are not listed as colloids. So the point behind these two problems is to help you understand the concept of dialysis, which you'll probably use as a nurse, where you clean somebody's blood based on things that can fit through microscopic holes or diffuse through microscopic holes, and things that are too big to diffuse through the microscopic holes in the dialyzing bag. And the things that are too big to fit through those holes are called colloids.